Okay, good afternoon, everybody. So I'm going to give a lightning talk so that we can all continue with the schedule. And I'm going to talk about what I call the spectral component analysis. And as you may guess, I'm just trying to build on the story that we developed yesterday, and I'm just going to extrapolate things a lot, so just bear with me. This is still in a very preliminary phase, and I want to talk about peeling off chemical layers in near-infrared spectroscopy. So, short introduction, what I want to do is just present an idea how to use real spectra, real measurement data to describe and create principal sources of variance. So not components that are calculated, but components that are actually measured. The background, as I just said, is the subtraction of the closest solvent spectrum. So instead of just doing solvent, we're going to do other components. So the precondition for this is that some information, chemical information about the sample that you're measuring is known beforehand. And I will show that the application, what exactly I'm talking about, is mostly in modeling complex liquid systems. And I'm going to give an example of human urine, but other models like cellular and molecular biology are also applicable, fall into this category. And I'm just going to do a comparison with principal component analysis, and that's why I intentionally wrote the title as Spectral components so that I can facilitate that similarities and differences. So short introduction, but I work in a molecular cell biology lab, and we're applying aquaphotomics, and we're trying to bridge the gap between spectroscopy and water biology. We have the molecular biology lab that I work in is concerned with the aquaporins, which are water channels discovered by Peter Agre in 2004, and we know that we have them in our body and brain, and we know that we have them, but still we don't know what exactly they're doing and how we can utilize them in therapy, and how we can make drugs to solve problems like edema and things that are related directly to water transport. So in order to address the entire water biology work, we worked on a different scale. So we started looking at water and ions, and then now currently we're looking into small molecules. We're working with monosaccharides, sugars, and amino acids, and we're having some nice success there. We're expanding that to polymers. We're working with small polypeptides, and what I'm going to talk about today is the application in biofluid, exactly the near-infrared urine measurement, and we're at the higher level. We're doing cell experiments, cell near-infrared measurements, and tissue. We're working on a human skin measurements specifically. So out of all this, let me focus on biofluid and urine measurements, and one of the things that we're after is trying to diagnose ovulation from human urine. So there are many ways to do that. The most preferred by many researchers is the tracking of luteinizing hormone. Other hormones are possible candidates, but mostly LH is the one that is preferred because it does have a really sharp peak around the 14th day of menstrual cycle. However, as sharp as this peak is, it's not at all easy to catch because of this slide. This is a picture from Wikipedia where you can see the variation in the luteinizing hormone. This is the intensity in standard units. These are ELISA units, and this is the day of menstrual cycle. So this is bright area is the variation in age, so when we're young and when the ladies get older, and this is the variation within one person, so this is inter-cycle variability. This bright inter-woman variability is a bit wider, but you can see still that it's not day 14 or 15, it's somewhere between 7 and 20, 22, 23, which is still a large amount of variation, so it's not easy to catch. And of course there are differences between people, but intercycle within just one person we have a larger variability. Anything that you do with urine also falls into the category of metabolic disturbances, so really there is a lot of variation. And here this is the kind of a summary of what I'm talking about, chemical summary. This is on the left here, low molecular weight components of urine, and these are high molecular weights of urine. And out of low molecular weights, almost 56 or 58% of dry mass of urine is composed of urea, and there are all others. There are iron, sodium, potassium, and chlorine, phosphate, sulfate, and other. So this is the biggest part. There are smaller, some proteins, albumins, and cholesterol, other components, but these are let's say, concentration-wise, lower components, lower grade constituents. So in order to understand, I want to try and decompose by using this entire chemical mess by using near-infrared spectra. So what I did is we took a lot of near-infrared spectra, and what I'm trying to show you here, hope you can see this, in cyan, in blue, light blue, I just took the spectra of urine and I just subtracted the closest pure spectrum of pure water. And to understand what that means, I also took a spectra of just pure urea solution at 1 molar and in this magenta. So what I want to argue here is that most of the variation in urine when pure water is removed comes from urea, which means that these spectral patterns are very, very similar. Because of that, I didn't subtract. I wanted to add something, not just subtract pure water. So I made different solutions of urea, starting with 100 millimolar, 204, 8,000, and 3,000. And then I induced temperature variation in all of those spectra, and then I subtracted the closest spectra of urea. And once I did that, the result that you see is here, given in light blue. This is urine minus closest urea at whatever concentration. I'm keeping record of that, at what concentration and what wavelength, of course, but I'm not going to focus on that. This is going to be, all of that is going to remain recorded in that future model that I'm trying to build. So exact position and all the characteristics of the pure water spectrum and of the urea spectrum. But what I want to show is, I would like you to notice how close this urea subtracted spectrum is to just sodium chloride that's in water. This is 100 millimolar. The spectral pattern is very, very similar. You could even be fooled that the blue spectrum is also sodium chloride. Of course not, because there is some very, very slow intensity bands that are not urea. This is CH and NH region. And here, those are the bands coming not from urea, but other NH carrying identities, such as proteins and everything that we do have. Like it is this circle. So there is a variability left, but after you remove urea, you get a much, much better picture. And what's left is sodium chloride. And if you look at sodium chloride and potassium, it's very similar. So you get like 8% here and 3, and with the chlorides you get 14. So you get about 25%, which is the half of urea. So the next, what this suggests is the next largest components are ions and solute, which by looking at this chart makes sense. So my argument here is we can subtract urea and then we can subtract pure water, and in that way decompose spectrum of urine into pure water, urea, and whatever is left, this can be also subtracted, and whatever is left in this difference. And then we can gradually reduce the complexity of the urine spectra and improve the understanding. So this brings me to my last slide, and the purpose of this method is this is what I'm trying to do. This is my personal view of how I see chemometrics nowadays. It's generally, which is good, it's shifting towards mathematics, but we're kind of losing track of chemistry, and it becomes increasingly difficult with the increase in mathematical complexity to reinterpret that data. So we're getting away from chemistry, but we're replacing that complexity with the hundreds of mathematical parameters that we equally know nothing about. So I don't feel like we're really making big progress. We need to change something, and this is still not the answer, because it's not a method, but it's what I'm thinking about. So I want to bring chemistry back into chemometrics and to use the knowledge that we can get, that is available, and use it to build better models. And what this method, what this example is I'm trying to show is that it facilitates reinterpretation. Once you subtract urea, you can directly assign whatever the variance is to urea. So there is no thinking, there is no, you don't have to crack your head about what's happening here and what's happening here. You do after, when you remove the salts, then you need to start a thing, but that's a very, very small part. The most, diagnostically, the most important part, but interpretation is much, much easier. So in this slide, this is my last slide, so I'm gonna compare what I call a spectral component with principal components. So in spectral component analysis, components are real measured spectra that are pre-generated, they're pre-existing. And in principal components, they're calculated, they're generated from sample. That's what statistic is doing. We're trying to learn from the sample. Here, we're pretending we already know something, which partially is true, and there is no perfect way. In this case, when we generate from the data, if the data is noisy, that's what we're gonna get back. So we're gonna get some level of disturbance. Potentially, if the components are pre-generated and the spectra are not noisy, they're recorded well, this might give a little bit of robustness towards some outlying sample, and then it's kind of a gray zone. Is it a good thing? Is it a bad thing? Sometimes we can protect, so we can sometimes build better models unless the outlying samples are actually the ones that are carrying the important diagnostic information. So it's a trade-offs here, but that's what I'm just comparing here. Another thing is that these components, because you know what you're measuring and you know what you're subtracting, they are chemical. They're not mathematical, so of course the mathematics comes from the sample. It doesn't come from thin air. So I'm just want to try to remind you that it's constantly a trade-off here, but I'm just kind of shifting the balance back to chemistry. And the thing that I like about this is that these components are correlated. That's my that's my biggest source of thought when I work with the principal component, all the matrix decomposition, the multivariate methods. What they came, if you go back a couple of decades, PCA came actually from psychology. That's where all these methods originated. And they were tailored for psychology. And in psychology, you have these different tests that are taken, given differently designed, and they're given to people. You just fill out the form, and that form is scored. So those tests are not uncorrelated because they're designed to be that way. Every spectrum is heavily correlated. You can take all these variables there. So correlation is a huge issue. So if we have a correlated, if we remove some part that is done in a correlated manner, my point is that I consider that to be beneficial. And so once we remove correlated, what we're going to be left, the residuals are potentially going to be uncorrelated or less correlated. So yeah, one of the most important thing is that in PCA, the not correlation, the lack of correlation is assumed, which is essentially not true. Anybody who's taken a spectrum knows that the variables are really correlated. So I hope that this will just bring some new, fresh thought ideas into how we deal with spectra and how we treat them in the future. I will certainly work with a lot on this. My current, since I generated a huge database of urea spectra, as Bernard asked quite nicely yesterday, so I'm trying to decrease. I used several concentration levels, but I'm working on decreasing the workload that is required in generating such a database, because for everything you have to do concentration gradient and you have to do temperature gradient, and that takes time. So I'm looking into ways to cut that workload and then to make that database creation as easy as possible, which will in turn make it possible for people to include more chemicals and more perturbations and more inquiries into the sample that we're looking at. So this is urine. There are other samples. One of the samples that we're working with are cellular models. We're taking fiber spectra of the cells in a petri dish, or we're taking metabolites. So with the metabolites, we already know the fluid that we're supplying to cells, so that is already a component that is somehow either chemically known, or it can be. We can take spectra and we can inquire into that. So all these types of things that we can get at least one or two pieces of information can be treated in this way, and I hope it will be helpful. That would be all. Thank you for your attention, and I hope I will be happy to take any questions that you have. Thank you.
Thanks, Dusan, for having been very strict on time. We have time for some questions, so if anyone has. I have just a quick comment, especially in the domain of MCR analysis and infraspectral, whatever kind of application. It's becoming more and more popular to let's say, use what in machine learning is called dictionary search. It's something similar to the concept that you are elucidating here, because it's the idea that you gather spectra from known sources, and then instead of looking for components like being optimized by the software, you're just trying to search among these possible lemmas in the dictionary that would be like spectra of known origin that you register in terms. Have you had a look at that, or maybe you may have a look at that in the development of this kind of approach?
Yeah, thank you for the comment. I'm not familiar with dictionaries, but I am looking into that. Actually, the optimal solution, in my opinion, is going to be some hybrid method, because the problem with the temperature composition and all these is that there are, especially if you go to the low-level information, small bands, weak bands, the non-linearities kind of take over. So that's the problem that is difficult to resolve in mathematical methods. So I'm hoping that going halfway and supplying some of that nonlinear nature to the software will help it make better predictions. So in the end, it's going to be a very small database, small enough that to enable reasonable average mathematical methods to perform better.
Okay. Is there any other question or comment? So if not, we thank once again Dusan and all the speakers of today's session.
Thank you.
And I leave the floor to the next chairperson for the poster session, for the poster pitch.